in the last video, we ended up talking about what a receptor is. And so we need to talk now about receptor-mediated endocytosis. So when you have a cell, and on the outside of the cell, you have this receptor. Something is going to be able to bond to this receptor. Let's say it's some type of hormone. And what happens then is that this hormone is able to activate the receptor. And the cell starts to go through endocytosis, which means that the cell membrane actually begins to fold inward, and it brings that receptor with whatever chemical is bonded to it into the cell, until you finally see this vesicle with the receptor inside and that hormone. Now remember, anytime you pinch off a piece of the cell membrane, so what's going to happen is these ends of the cell membrane will start to come together until you pinch off a piece of the cell membrane and you have the receptor with the chemical on the inside. This cell membrane that's been pinched off is, if you remember, what we refer to as a vesicle. So look at the overhead just for a second, and you can see again in orange, red, whatever color this is, these are the receptors in the membrane, and then this membrane is folding inward until it pinches off, and the receptors are on the inside. So here's an actual cell where you can see the receptors on the inside here, the little black dots, and then this cell is bringing all these receptors in the process of endocytosis, so that whatever was bonded to that receptor, which is what the important thing is, gets inside the cell. Now the other weird thing though is that the receptor also gets into the cell. So the question is, why would you want to bring those receptors into the cell? And then next question is, how long do they stay in the cell? Do they ever get out of the cell again? What's the benefit to the body? What's the physiological benefit to our body to have these receptors along with the chemical go inside the cell? Well, one of the major benefits is now, if this receptor is on the inside of the cell, is the receptor able to respond to anything that's going on the outside of the cell? And the answer to that is no. The cell actually, if it pulls its receptors in, it's not able to respond, the cell doesn't respond to the outside world as well as it did before. And you think, well, that might not be such a benefit, but in actuality it is. It's a big benefit to the cell. And the reason it's a benefit is, let's say for instance, maybe this hormone that's out there is a type of hormone that we release when we're under a lot of stress. So maybe you have a crazy college student taking a, I don't know, biology class, and they're super duper stressed out. And so now the cell is responding to this chemical. When this chemical bonds to the receptor on the cell, it makes the cell do something. And usually in a stress response, it speeds the cell up. It makes the cell produce more of its other chemicals or do whatever its processes are faster and faster. So if I'm under a lot of stress, but I can suck those receptors to the inside of my cell and have those receptors stay there for a while, do I feel as much stress? And the answer is no. I actually don't feel as much stress as I did before. So that now it can be a benefit to me if I'm in a stressful situation to get this receptor in by receptor-mediated endocytosis, get the receptor in, and then my cell basically goes on a holiday. It can't feel the stress, it can't respond to the stress. And anytime receptors go into the cell and the cell can't respond, We call this down regulation. When the receptor finally comes to the outside, and it will eventually, okay, hopefully, come to the outside. Nobody knows how that actually happens or what makes it go to the outside, but when it does, when this receptor pops back out again, this is what we would call upregulation. So this is the process of receptor-mediated endocytosis. The last process that I want to go through with you is something called exocytosis. And exocytosis occurs when you want chemicals to leave the cell. So endocytosis, we're bringing things into the cell. Exocytosis, things are leaving the cell. So in exocytosis, let me just kind of erase this stuff right here real fast. We're going to have the opposite event occurring as endocytosis. And let's say I have a cell, and maybe inside this cell I've created some protein. I don't know what protein it is, but I've created a protein. Now, proteins are very large molecules, and they can't get through those gates. They can't get through that phospholipid bilayer. They're too big. But I need this protein to leave the cell for whatever reason. Maybe the protein is an antibody. Antibodies are proteins that we make with our immune system, and these antibodies help us to fight diseases. So I need to get it out of my cell so it can help to keep me healthy. So the first thing I'm going to do is I'm going to take this protein, and I'm just going to put a big P here for protein, makes it a little bit easier for me. And I'm going to put this protein into a vesicle. I'm going to wrap it with a basketball-like structure of my phospholipid bilayer. And then as soon as I do that, this vesicle is going to float to the surface. And so now this is made up of cell membrane, which is the same thing as the cell membrane on the outside of my cell. So now I have the cell, and I have the vesicle right up along the cell membrane with, of course, my protein inside. So that the cell membrane from the vesicle and the cell membrane of the cell itself will start to kind of dissolve into each other, you might say. And you'll get something like this, where part of the vesicle becomes part of the cell membrane. And now my protein that was in here is able to leave the cell. This is the process of exocytosis. Now, eventually what will happen is my cell is going to get a lot bigger because my vesicle will become part of this membrane. And so it makes the cell a lot larger as well in the process of exocytosis. But this is how our cells allow things that are too large to pass through gates or too large to pass through the phospholipid bilayer leave the cell. So if you look at the overhead here, in the top picture, this is the process of endocytosis where things are going into the cell, and notice we're pinching off a vesicle. Whereas in exocytosis, things are leaving the cell, and the vesicle actually becomes part of the cell membrane. Same thing in this picture here. You can see the vesicle fusing with the cell membrane and then becoming part of the cell membrane, and whatever's inside that vesicle is leaving through exocytosis. So any questions? Okay, so we have been talking about the cell. And as we talk about the cell, I told you that we're going to go through it kind of in steps. We are talking about the cell membrane first, and we've discussed passive transport, we've discussed active transport, endocytosis, exocytosis. Now what we're going to do is talk about the inside of the cell, and we're going to talk about the different organelles that are in the cell. So one of the first things that I want you to know is that our cells have particular shapes. And so normally when somebody draws a cell, they usually draw it round, but we actually don't have cells that are round. We have cells that sort of look like cubes, believe it or not. And we have cells that kind of look like little eyeballs. We have cells that are very tall, look like rectangles or columns, okay? And these cells have to have very distinct shapes. In order to hold the shapes, we have proteins that develop right underneath the cell membrane. And these proteins are in a kind of particular structure, and they help to hold the shape of these cells, and these proteins are called the cytoskeleton. And the cytoskeleton are made up of proteins called microtubules and microfilaments. But these are particular proteins, and their main job is to help to hold the shape of our cell membrane, okay? Now, we have other types of proteins inside our cell. We're going to talk about these as we go along. These are proteins like actin and myosin. These proteins, we'll talk about the fact that they help us to move the cells. They actually create an environment where the cells can do movement, like for instance, the cells of your muscle, they're able to move, so therefore your whole muscle moves. And this is because of these different types of protein like actin and myosin. So microtubules, they also do a couple other very interesting jobs. And again, we'll talk about this as well, but microtubules help to move things inside the cell. So you can actually see, there's some really cool YouTube videos on microtubules, and you can see microtubules can actually do this movement inside the cell, almost like walking, and they can grab onto something like they're holding it on their back, like a backpack, and they can walk around in the cell, and they can move things from one area of the cell to the other. So microtubules are pretty interesting little proteins. Now, on the outside of our cells, sometimes we'll see things like this. So maybe you'll have this rectangular looking cell, and then instead of the top being flat, it'll have this kind of structure here, and this is what we would refer to as cilia. It almost, because they're microscopic, they're really small, so it almost looks like there's little hairs on the outside. But you can see in the overhead that these are cilia here, and we say that they look like little finger-like projections from the outside of the cell membrane. And down here, these are little network of things. These are proteins. These are the microtubules that go up into the cilia, and they look like this. So here are the microtubules that are inside the cilia. Here's the cilia here. And proteins have the ability to change shape. And because the proteins change shape, they allow the cilia to change shape. Same thing with flagella. Now, flagella is something that we find on a sperm, okay? So a sperm has like a little tail, and this is the flagellum, okay? And inside here, very similar to cilia, it has these microtubules that allow it to move back and forth so it can do its swimming thing. Now, cilia, okay, they all beat in one direction. Okay, so for instance, we have cilia in our trachea here. And this cilia all beats upward, constantly moving upward in one direction. Now the reason the cilia is beating or moving upward is because anytime you inhale like dust or other particles, the cilia is there to help push it out so it doesn't end up in the lungs. Or maybe in the female reproductive tract, we have some cilia in the tubes that connect the ovaries to the uterus, the fallopian tubes. And those cilia beat in one direction to the uterus so that the egg that leaves the ovary can be moved down the tube and into the uterus. So in this overhead right here, this is the cilia, all beating in one direction. This is in the trachea, and it's moving mucus, this mucus-like stuff that catches the dust particles and helps to move it out of our trachea. And we swallow it all day long, don't even know it's there, so that we don't get all the dust and everything building up in our lungs. So, although I told you we're going to talk about organelles, I just wanted to finish off talking about some of the different structures that can affect the cell membrane. So let's talk about organelles now. And the first organelle that we're going to go over is a type of organelle that is called a centriole. Now, organelles, they are found inside the cell. So remember, we talked about this already, but just kind of to refresh your memory, we have a cell, and the fluid in the cell is the intracellular fluid. So our organelles are inside the intracellular fluid, and then of course the fluid outside the cell is the extracellular fluid. And cells are bathed in fluid on the outside. So this intracellular fluid, okay, so we have the intracellular fluid, the ICF, okay, and we have organelles located, organelles, it's an A, located inside our cell. These two together form what we call the cytoplasm. So the cytoplasm has the intracellular fluid, it has the organelles. Now the intracellular fluid, let me just tell you, this is not a, hang on just a second, my overhead keeps popping in and out. The intracellular fluid, this is not a watery substance, it's more sort of a gel-like substance, kind of the consistency of, let's say, egg whites, okay, thicker like that. And our organelles are inside this gel, and we have two types of organelles. We have organelles that have a membrane, and then we have organelles that don't have a membrane. And the first organelle, the centrioles, these fall under the non-membranous. And the job of the centrioles, okay, these are found in our cells, in animal cells. And the job of the centrioles is to help to divide our cells. So the process of dividing cells, which we'll be going over in a couple lectures, is called mitosis. And so you get these centrioles, and you get one pair, let's say, on the north pole of the cell, and one pair on the south pole. And the centrioles are able to produce microtubules. So there's these long proteins that go in between, sort of looks like a bunch of clotheslines, in between these centrioles. So let's say that this is a cell, and you have a pair of centrioles. So you're going to have them looking kind of like this. They line up sort of like this on each end of the cell. And then in between these centrioles, they produce these microtubules. And these microtubules allow our DNA to line up on these microtubules. And then what the centrioles are going to do is they're going to pull, literally, they're going to pull on the microtubules. And as they pull, they're helping to pull DNA to one side, DNA to the other, and they're helping to pull the two cells completely apart. So the centrioles are necessary to pull all of our cells apart so that we form not just one cell, we form two new cells, or we go from one big cell to two daughter cells, and we can only do that with the centrioles. So you can see the overhead picture right here. This is a pair, this is a pair of centrioles. Okay? The next very important organelle is called the rough endoplasmic reticulum. That's a big one. So this is the RER. This is the rough endoplasmic reticulum. And the rough endoplasmic reticulum has two major jobs. Okay? Job number one. Stores calcium. So the rough endoplasmic reticulum has some hollow regions. And so you can see this is it right here. And it's hollow, and it has the ability to store calcium. Now, when scientists were looking at cells, and they finally had a microscope, and they were able to look on the inside of cells, they saw an endoplasmic reticulum, which is right here. This is the smooth one on the outside to the right. And then you have the rough one, which has all these little kind of pink-looking dots. Now, we don't know what color it is. We can't see that with a microscope, so they've just used some artistic thoughts to put the little pink dots on here. But because of the little dots, they called this the rough endoplasmic reticulum because it had no dots. This is the smooth endoplasmic reticulum. Notice also this is part of the nucleus of the cell. And the rough endoplasmic reticulum is very large. It goes from the nucleus of the cell all the way to the cell membrane. And that rough endoplasmic reticulum, again, stores calcium, but it also plays a part in protein synthesis. Now, what the word synthesis means is to build or to make. So this is the rough endoplasmic reticulum playing a part in building or making proteins. Now, it turns out that all those little dots that were on the rough endoplasmic reticulum were also an organelle. And this organelle is the ribosome. The ribosome works along with the rough endoplasmic reticulum in protein synthesis. So we're going to talk about protein synthesis in a little bit, but the ribosome helps with this protein synthesis. Now, the ribosome has two parts to it. So you can see in this picture here, there's a large and a small part. These actually remove from each other or come apart. The large one is called the large subunit or the large ribosomal subunit, and then you have a small ribosomal subunit. Okay? This is another picture of the rough endoplasmic reticulum. You see all the little dots here? Those are all the ribosomes. Okay? So now let's talk about the smooth endoplasmic reticulum. So like the rough endoplasmic reticulum, the smooth also stores calcium. But unlike the rough, it doesn't have anything to do with protein. It actually has more to do with fat. And so the smooth endoplasmic reticulum produces phospholipids. And remember, those are the fats that make up our cell membrane. It also produces fatty acids, which help to make up the fat of our body, like the triglycerides. And then it also helps to produce anything else that's made out of fat, like maybe hormones, okay? And which ones are those? Right, those are the steroid hormones. They are the ones that are made out of fat. So this is what our smooth endoplasmic reticulum does for us. The next organelle is the Golgi Apparatus, named after an Italian scientist, Dr. Golgi. And the Golgi Apparatus has two major jobs. Job number one is it is responsible for creating vesicles. So anytime we go through endocytosis or exocytosis, these vesicles are being made in the Golgi apparatus. And this is called vesiculation. The second major job is it creates glycoproteins and glycolipids. Now these are, if you remember, we talked about what glycogen is. And glycogen is a big molecule. It's made out of tons of single tiny glucose molecules. So you take hundreds of glucose molecules and you bond those together and you get glycogen. Okay? If you take that glycogen and bond it to a protein, you have a glycoprotein. If you take that glycogen and bond it to a phospholipid, you have a glycolipid. So glycogen can be attached to proteins, it can be attached to a phospholipid, and the Golgi apparatus is responsible for doing that. So if you look at this overhead right here, this is the Golgi apparatus, and notice it is forming vesicles. That's what these little bubble-like structures are. These are the vesicles. This is actually an electron micrograph, which is a microscope that can blow something up like 200,000 times its normal size, and that's what this is right here. And so you can see the little bubbles that are coming off. Those are vesicles coming off of this Golgi apparatus. So sometimes you'll hear people talk about the fact that this is the post office of the cell. It actually can create a vesicle, and then what's inside that vesicle? Well, a lot of times there's protein in the vesicle, so we're going to take the protein and put a glycogen on it before it gets shipped out. So it's almost like you're stamping the vesicle, okay, with that glycogen, sort of like a glycogen flag being added. So this is just a picture showing you the vesicles are coming through the Golgi apparatus, and these are the proteins inside, and then exocytosis occurs. All right, so the next organelle is a lysosome. And many times you're going to hear people call lysosomes the garbage cans of the cell. the job of the lysosome is it releases digestive enzymes. Now, lysosomes, what they're able to do is they're able to move throughout the cell and find garbage in the cell, okay? So anytime our cells go through some type of chemical reaction, there's always going to be a waste product, always. And so that waste product is typically cellular garbage. So the lysosome moves through and can grab a hold of whatever this garbage is and suck it in to the lysosome, into itself, sort of like putting it in the garbage can. And then once it's inside, it releases these enzymes to tear the garbage apart, so that it's almost sort of like a trash compactor. So if you look at this picture here in the overhead, this is a lysosome, and this little white thing is supposed to represent the garbage. And notice the lysosome has like these little arms. It goes around it, almost like phagocytosis, and it engulfs the garbage. And then once this waste product is inside, the lysosome releases its little digestive enzymes and destroys the waste product. And notice it's compacted that waste product. Because if a cell has too much garbage inside of it, it can't work, it can't function appropriately. It's kind of like, I don't know if you've ever seen those shows where you have hoarders, okay? And you have these people who have so much garbage inside their house, literally garbage, that they can't get to rooms. They can't live in their house, and it's filthy. And so it's the same thing with our cell. If our cell accumulates too much garbage inside, the cell can't function, it's going to die. So the lysosome helps to prevent that garbage buildup from accumulating and happening. All right, the next cell, the next organelle, I should say, is a peroxisome. And peroxisomes secrete an enzyme called catalase. Remember, if you have ASE at the end, this tells you that this is an enzyme. So peroxisomes release catalase. Now here's what catalase does. Catalase takes this chemical, which is a waste product, H2O2. This is hydrogen peroxide. We make this in our cell as a waste product. And catalase breaks it down into oxygen and water. 1/2 O2 just simply means a single oxygen atom. Okay? So catalase can take hydrogen peroxide and break it down into oxygen and water. Now, one of the questions is, why is it that we need an organelle that is there just simply to break down hydrogen peroxide? Why do we have this one organelle making this one enzyme to break down this one waste product? Well, hydrogen peroxide in high concentrations is actually very explosive. So could you imagine hydrogen peroxide building up and building up inside our cell until it explodes? It would not be a good idea. Yeah, that would be a very bad idea. So we want to make sure that this waste product gets torn down as quickly as possible. And so that's what catalase is for. All right, the next organelle is an organelle called the mitochondria. So the mitochondria, maybe you heard somebody say this is the mighty mitochondria, the powerhouse of the cell. Well, the mitochondria is where we produce the majority of our ATP molecules. This is our adenosine triphosphate. And if you remember, we talked about the fact that you have to take a phosphate and add it to a protein in order to get that protein to move, to get it to change shape. And so ATP can make a lot of proteins move and change shape, and our body can do things. So this is why we consider this an energy source, because it has the ability to change the shape of proteins. So the mitochondria, which we're going to talk about, is where most of the ATP production is made. Now, cells that have mitochondria can be plant cells, can be animal cells. Now, when mitochondria, I should say where ATP is made. When ATP is made in plant cells, we call this photosynthesis. When ATP is made in animal cells, we call this cellular respiration.